Let's talk about something called genetic fingerprinting, which is used in the field of forensics. Remember that gel electrophoresis is a machine that we're going to use to separate fragments of DNA. And these machines separate this DNA based on their molecular size. Within eukaryotic DNA, we can separate things called STRs and VNTRs. STRs are short repeated sequences of DNA that don't code for a protein, while VNTRs are basically the same thing but larger. And each person's going to have a specific amount of these STRs and VNTRs. These repeats allow us to separate the DNA based on length. So because essentially every person has a unique set of these repeats, we can use it in a gel electrophoresis machine. And this was discovered back in the 1980s, and it's a major tool still used in forensics today. What we basically do is we compare these repeats between crime scene DNA and people that we think are suspects. Let's go over an example. You can see specific fragments matching up with each one of these people that are suspects. However, suspect 2 matches every single fragment from the crime scene. So in this case, suspect 2 matches the crime scene, and it doesn't mean that they automatically did it, but it does mean that they were found at the crime scene. So we can use this tool to identify suspects and if they were actually at a crime scene or not. And we can also tell the paternity of a child using the same method, because each person is going to inherit half of their fragments from mom and half of them from dad. So if you look at the fragments that the child inherited from the father, not the mother, we can tell that dad 3 is this child's actual dad.